Good day, this is Professor Will, CBMD PhD, and today is Monday, September 27, 2021. It is 1:24 and 52 seconds a.m. Eastern Standard Time, and today's research, genetic brain disease. So let's hop to it here. A genetic brain disease reversed after birth. Date September 21st, 2021. Source Riken. Researchers report that Kleistra syndrome, a genetic disorder that leads to intellectual disability, can be reversed after birth in a mouse model of the disease. The series of experiments showed that postnatal treatment resulted in improved symptoms, both in the brain and behavior. Normally, we get two good copies of both genes, one from each parent. In Kleistra syndrome, one copy of the EHMT1 gene is mutated or missing, and this leads to half the normal amount of GLP, a protein whose job is to control genes related to brain development through a process called H3K9 methylation. Without enough GLP, H3K9 methylation is also reduced, and the connections between neurons in the brain do not develop normally. Researchers still don't know if Kleistra syndrome is a curable disease after birth or how this epigenetic dysregulation leads to the neurological disorder. Research studies in mice have provided new information about what causes the behavioral abnormalities associated with the syndrome and have shown that a cure is a real possibility in the future. Reasoning that extra GLP might be an effective treatment, the researchers performed a series of experiments in mice that were engineered to have only one good copy of the EHMT1 gene. The brains of these mice show characteristics of the human condition, including 40% less GLP and 30% less H3K9 methylation. The mice also display several behaviors seen in humans with Kleistra syndrome, such as reduced locomotion and greater anxiety. After each experiment, the researchers measured these factors and compared them to normal mice to see if the treatment had been effective. The researchers artificially induced GLP production after birth in two experiments, one throughout the whole brain and one specific to adult neurons in the brain. And treatment in 3 to 4 week old juvenile mice quickly rescued GLP and H3K9 methylation levels in the brain in both tests. Behavior improved several weeks later, but only when GLP was increased throughout the whole brain. Thus, brain and behavioral symptoms were successfully rescued by treatment that increased GLP levels throughout the brain after mice had already become juveniles. The researchers next wanted to know why their treatment only fully worked when GLP was increased throughout the brain, not just in neurons, and thinking that perhaps the disorder abnormally activates microglia cells in the brain, which are known to control immune responses, such as inflammation. The team searched and found a well-known inflammatory response in the brains of the model mice, along with greater amounts of activated microglia cells. Knocking out a key protein involved in the inflammatory response reversed some of the brain abnormalities caused by inflammation, but did not change the behavior. And this means that brain inflammation is only part of the story. To have a complete understanding of the disease, we need to find out what happens in other non-neural cells when we increase GLP. As this is the first report of neuroinflammation in Kleistra syndrome, the next step is to find out if it also occurs in the human condition. Researchers believe the chances are high and say they would not be surprised if other neurological diseases caused by epigenetic deregulation were also related to abnormal inflammation in the brain. Although researchers don't yet know if their findings will be applicable to patients with Kleistra syndrome, researchers have shown that a cure after birth is possible and believe this will bring hope to patients and their families. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.